Hello all, welcome back to our YouTube channel Botany by Sandhya and Varshni. In this video, let's see about an interesting topic called meiosis. Okay, let's get started and check it out. Have you ever wondered why children look different from their parents and why two siblings even vary from each other? The answer lies in a process of meiosis. This meiosis generates the gametes for reproduction. Meiosis is a process in which haploid cells are produced from a diploid cell. For this process to occur, the chromosomes should be correctly sorted and distributed in a manner to create genetically unique cells. Meiosis occur in special cells called germ cells, which present inside the gonads. That is the sex organs of both males and females. For the successful formation of the new haploid cells, two rounds of divisions are necessary. These two rounds are termed as meiosis 1 and meiosis 2. Actually, the process of mitosis and meiosis looks similar, but there are few major differences. The first difference is mitosis results in two diploid daughter cells, whereas the meiosis results in four haploid daughter cells. Like mitosis, the meiosis also begins after the cell completes the G1 phase, S phase, and G1 phase of interphase. In G1 stage, all the cellular contents excluding the chromosomes are duplicated. In the S phase, each of the 46 chromosomes is duplicated by the cell. In G2 phase, the cell double checks the duplicated chromosome for any error and it makes any needed repairs. After this G2 phase, the meiosis occurs, which is occur in two stages, that is meiosis 1 and meiosis 2, followed by the cytokinesis. Now, as a result of duplicated chromosomes, sister chromatids are found in a cell and a pair of centrioles in the cytoplasm, and it began extending the microtubules which will form the meiotic spindle. This initiates the meiosis 1. In meiosis 1 take place in four stages, that is prophase 1, metaphase 1, anaphase 1 and telophase 1, which is followed by the cytokinesis. After the cytokinesis, meiosis 2 occurs. Okay. Let's see the prophase stage in detail. The prophase occurs in five stages, that is leptotin, zygotin, pacotin, diplotin and diakinesis. In leptotin stage, homologous chromosomes condense to each other. Next is zygotin stage. In the zygotin stage, the homologous chromosome of sister chromatids lie side by side in a process called synapsis, forming a tetrad. The homologous chromosome looks similar, but it does not have identical genes. This zygotin stage is followed by the pacotin stage. In pacotin stage, the crossing over occurs between the pair of chromosome to form the chiasmata. The chiasmata is mentioned in the diagram. After this diakinesis takes place, the homologous chromosome continue to separate and the chiasmata move to the end of the chromosome in diakinesis. The diakinesis is the end of the prophase 1 stage. This diakinesis is followed by the pro-metaphase 1. Pro-metaphase 1 is the stage in which the spindle apparatus formed and the chromosome is attached to the spindle fibers by the kinetochores. Okay, the nuclear membrane actually breaks and the kinetochore invade the nuclear space and attach to the kinetochores. Metaphase 1. During this stage, the bivalent arrange as a double row along the metaphase plate. Due to the independent assortment, the alignment may vary, but it leads to the genetic diversity. Anaphase 1. The homologous chromosomes separate and move to the opposite poles of the cell. This is followed by the telophase 1. The chromosome decondenses and the nuclear envelope reforms. This telophase further leads to the cytokinesis, where the cell division occurs. The cell division occurs to form two new cells. As a result of meiosis 1, two haploid daughter cells are formed. So now meiosis 1 is completed, followed by the cytokinesis, and two daughter cells are formed. This meiosis 1, after this meiosis 1, meiosis 2 takes place. Meiosis 2 is similar to the mitosis 1. Here, these four stages occur like prophase 2, metaphase 2, anaphase 2 and telophase 2. In prophase 2, the chromosome number doubles In metaphase 2, the pair of sister chromatids line up at the metaphase plate. In anaphase 2, the pair of sister chromatids separate due to the law of independent assortment. In telophase 2, the chromosomes are fully separated, followed by the cytokinesis. As a result, 4 daughter nuclei are formed, which is haploid in nature. Okay, that is all about my meiosis. Let's see the significance of meiosis. Sexual reproduction leads to genetic variation among the offspring. Meiosis makes genetic variation possible in 3 ways, that is through crossing over, reduction and fusion of gametes, and independent assortment. That's all about meiosis. Thanks for watching. If you like this video, please do subscribe to our channel. And until next video, this is Varshni signing off.